So what happens in the next part of the digestive process? This is the major portion of digestion. So here we have the small intestine, right? And small intestine, this region that comes from the stomach, we call it the pyloric region. So if you ever see that term pyloric, that's directly connected to the stomach. As you're going through your journey of the digestive process, the various regions of the stomach, the pyloric region is the last region before it enters the small intestine. The small intestine itself is divided into several major sections. And the first major section we call the duodenum, or duodenum, tomato. And it's in this first section where a lot of things occur that are very important to the digestive process. In this first section, I mentioned before that the chyme, that's what the food's called when it comes out of the stomach, the acidic chyme something needs to happen to it, otherwise it might affect the walls of the small intestine. So in here, we have we need something. So if you were going to change up the chyme in such a way that it was no longer damaging the small intestine, what would you do? If you said that you might neutralize the acidic chyme by adding a base, that would be an excellent choice. So coming into the duodenum here, we have this duct, a tube. And this tube, one of them goes to the pancreas, and that's going to be a major focus here, and another part of it goes up to another organ. It's called the bile duct, and it's going to go up to, I should have done this in a different color. I'm doing the whole digestive system here in black though. So this goes up to the liver and the bile and the gallbladder. Okay, so this part here that goes to the pancreas. The pancreas is a very special organ. And so the pancreas has a lot of roles that we're going to talk about in grade 12, but we also talk about some of it here in grade 11. The pancreas releases certain hormones like insulin and glucagon that play a role in sugar metabolism. And the pancreas also releases sodium bicarbonate. And you might recall sodium bicarbonate is used as a base. So as a base, this is released into the duodenum. And so it interacts with the chyme and it neutralizes it, changing the pH back to 7, which is excellent. So that is one job done. So the pancreas neutralizes the acidic chyme. So in addition to neutralizing the chyme, the pancreas also releases a whole host of special enzymes. So let's look at the enzymes that it does release. One of the major enzymes that it releases is a protease. So proteases, as we learned before, will take proteins that were already broken down in the stomach into long polypeptides. So in entering the stomach, this would be 100 or 1,000 long. Coming out of the stomach, they'll be 5, 10, 20 long. And they come into the small intestine, and the protease, which is released by the pancreas down this duct, it will take these and break them into amino acids. So now it's broken into an amino acid. And let me actually put it in the right spot. So when it comes in up here, they're joined together. When it comes down here, they're individual pieces. So now this polypeptide has been broken into amino acid thanks to this protease. So we have the polypeptide on this side, polypep, and it goes to be broken down into an amino acid thanks to the protease. The other thing that the pancreas also releases is a amylase. So there's also an amylase that's released. I guess I should use a different color to help with this. So an amylase is released. And the amylase, what it does is it takes those polysaccharides. So we had a really long polysaccharide in the mouth, and they were broken down into smaller polysaccharides, and so they enter into the stomach. That was the last time they're broken down in the mouth, and then they are broken down into individual units of glucose. So our polysaccharide, polysac, small, so it might be trisaccharides, disaccharides. These polysaccharides are then broken down into glucose units by the actions of the pancreatic amylase, not to be confused with the salivary amylase. Another thing that will come in here are the, another color, okay, so we'll go with this color, are the nucleic acids. And so these nucleic acids are coming in, and they're being broken down by a nuclease that's released by the pancreas into the small intestine. And so you're getting individual nucleotides that are being formed in the small intestine. So looking at this, we have nucleic acids, and they're being broken down into nucleotides by nucleases. So all these things are happening in the small intestine. So there's one thing missing. There's one thing I haven't actually brought into play here, and that is the lipid. So the lipid's a little bit different. So let's just finish with the pancreas. So the pancreas, what does it do? The pancreas, it releases sodium bicarbonate to neutralize the acidic chyme. It releases a whole barrage of enzymes that break down the macromolecules into their individual monomers. That's why I started the lesson off with this concept. And the pancreas releases hormones, insulin and glucagon, that assist with the metabolism and the absorption of glucose throughout the body. So that's more of a grade 12 discussion. Okay. So we still have one thing that's missing, and that is the lipids. So lipids include oil and fat. And as you probably know already, when you mix oil and water, they won't mix. You can kind of make a colloidal solution, sometimes with like a salad dressing kind of thing, but eventually the oil and the water will separate. So if you want to break down fatty substances inside our body, you need some way of incorporating that lipid in our water-based environment. And so we have another organ, a giant organ, with the liver. And the liver is responsible for so many tasks. There's a lot of things that the liver is responsible for doing. And you'll learn about that when you read it in the PowerPoint. But one of the tasks I want to focus on, this is a terrible liver, doesn't look like that, is there's another little organ here called the gallbladder. And I should do that in green because the thing that it produces is kind of a greenish substance. And so the gallbladder and the liver, they join this duct that the pancreas uses to the hepatopancreatic, this duct here. It combines a tube coming from the gallbladder liver area and from the pancreas. And so the gallbladder, what it will do is it will release a very special substance. So I need a color for this. What are we going to use here? What have we not used? Purple? Okay, so our bile today is going to be purple. And so the bile is created by the liver, and then it's stored in the gallbladder. Right? So this is my liver, and that little tiny organ there is the gallbladder. And the gallbladder, it holds the bile until we're ready for it. So the bile, when digestion is occurring, is released by the gallbladder, and it joins and it comes into the same area. And what the bile does, it's like a detergent. If you understand how detergent works, you might not. I don't know how much chemistry you know. But the fat, so I'm going to use yellow here to represent the, I need a different yellow. There are a bunch of colors here. I guess I'll use this yellow again. Okay, so the fat, as it comes in, this glob of, so you got this glob of fat. So we need to take those globules and we need to break them from big giant fat globules into smaller fat globules. And so the bile, what it will do is it will help with that breakdown process. Okay, so let's just go to a different section of the page here. And I guess I'll go over here. Let me just erase this. Okay, let's just go down here. All right, so the bile. So here's your big flat globule. And the bile, they're like little tiny salts. And the bile, what it's made up of, it's made up of this one end that likes fat. So it has a fat-loving end, fat-loving. And it has another end here that likes water, so a water-loving end. So it has two ends. These bile salts have a very special name. They're called amphiphilic. Amphiphilic, which means they like fat and they like water. They like both. And so what will happen is these bile salts, they'll attach to the fat globules. And this is just how detergent works. When you have oil or grease in your clothes, it tends to be greasy. Then you put in detergent, which kind of looks like this, and one end will stick into the fatty or greasy dirt, and the other one will be very hydrophilic and it'll like the water. And as you shake it around in the washing machine, it will pull the dirt out of your clothes. So then once this goes through this process is known as one of the key words I put on your sheet that you should know, emulsification. So in emulsification, what will happen is the fat globule will be broken down into smaller pieces, if I can use that word, of fat. And each one is kind of attached to its own bile salt, if you will. So now that this fat has been taken from a big giant fat globule where the surface area to volume ratio is not very good, to these smaller fat droplets, we'll call them, where the surface area to volume ratio is much higher, these in turn now, they can be better attacked and reacted by the last of my enzymes, which I'm going to call my lipase. So my lipase, what it does is it will take my triglycerides, my lipids, and it will break it down into fatty acids. Lipids. So the lipid digestion requires two processes. It requires the emulsification by bile, and then once you have these small droplets, these in turn are broken down into individual fatty acids. And then the fatty acids are then able to be digested. So they are digested. Now they need to be absorbed. And so that process is slightly different. So now all these things here happen in the small intestine. So in the small intestine, we had our polypeptides broken into amino acids, which I represented with the blue here. We have our polysaccharides being broken into glucose, which are represented by the reddish orange thing here. We had the nucleic acids being broken down into nucleotides, this kind of yellow gold. And then the lipid, this fat globule here, is emulsified by the bile coming from the gallbladder into small little droplets. And then those in turn can be broken down into fatty acids thanks to the action of the lipase. Okay? And then all those things will then keep on moving to the small intestine. So let's just continue our small intestine. Keep on moving along. And clearly not drawing it in the same way. So the wall of the small intestine, if I were to blow it up, if we go to the next section, so the next section of our small intestine, and remember I mentioned there's 3 different regions. The first one is called the duodenum. The duodenum is the very first 10 inches. And after that we get into the jejunum. And the jejunum is the next section. Can't spell very well here on here. I can't write. The jejunum is where the majority of the absorption occurs. Okay? So all these things have been broken down. If I were to blow up this wall right here, what you'd see is that it has all these folds. And what these folds do on the inner wall is they increase the surface area. And then if I were to blow up a certain section of this, so you can see what it looks like there, on those folds, you're going to see these tiny finger-like projections. And they're called villi. And once you look at the PowerPoint, you'll see a better representation. So this is not very legible. And so these little finger-like projections, they greatly increase the surface area of the small intestine. So what I'm now going to, so there's folds, and on the folds are villi. So what I'm going to do is I'm going to focus on the villi and what actually is happening in the villi. So what you need to understand is when we look at the small intestine, I look at it as a tube here, and all these nutrients that we just broke down, they're all flowing through the small intestine. It's going over all these folds, which are covered with all these villi. Okay. So as I go over those villi, so let me just draw a single villi here. And I like to draw it closer so you can actually see all these nutrients I've drawn here. So I'm just going to erase this section. Yeah, erase this section here. Get rid of this. Use that. So this is going to cooperate, eraser. I don't know what it's doing. Okay, whatever. Okay, so as I get to the villi, the villi is a finger-like projection. And in this villi is a blood vessel. So this blood vessel is coming in from somewhere, the circulatory system, which we're not talking about just yet. And it's coming into the villi, delivering oxygen. And as it leaves, it delivers oxygen, and we typically represent deoxygenated blood with a blue blood vessel. So we have these blood vessels here. And right in the middle of all this, there's another very special vessel. We call it a lymph vessel. Right? It's actually a lacteal. And so it joins another network. So down here, there's a couple of networks. One of them is the lymphatic vessel. And the other one is this blood vessel that's coming from somewhere. So I'm trying to keep this simple here. All right, so all right, and I'll just draw blue here. So we got these blood vessels, right? So we have one that's delivering oxygenated blood, one that's taking away carbon dioxide, and then we have this special lacteal. That's new. We've never heard that before, so I have to explain what's going on there. It's a new one. That's this gold yellow one. So let's take your glucose. So the glucose, what's going to happen is it's going to be sitting out here in the lumen, in that area of the open space of the small intestine, and it needs to be absorbed. So it gets absorbed into the blood vessel, and it gets taken away. So let's take the amino acids. The amino acids are now broken apart, and they are going to be absorbed and taken away into the circulatory system. Let's look at the nucleotides. So they're also floating around, and they're going to be absorbed, and they're going to be taken away. So all these small little units are now taken away, and they're brought through the circulatory system and distributed around the body. So it's in the small intestine, in the jejunum, where all this occurs. So all that from here just continues along the blood vessel, and it's all taken up. But the fatty acids are a little bit different. The fatty acids, they're big. Relatively speaking, compared to the other monomers, they're giant. They're very large, and they don't play very well, right? So the triglyceride was broken down to fatty acids, and they won't fit. So let me just do a slightly different diagram here to explain how the whole thing works. So if I use black here, I'm just going to flatten it so you can see what it looks like. That represents the villi wall, and below that we're going to have a blood vessel. So let's say this represents the blood vessel. And then below that, we're going to have this lacteal. It's another vessel that's going to carry away something else. Okay? So, can I make that smaller? I don't know. Okay, and let's make this bigger. There we go. So, as the various components here, once again, when I have my glucose, when it gets absorbed into the villi, it fits to these little tiny pores in the blood vessel and it gets carried off. Let's make that a bit longer so you can see it. Let's make it continuous. It gets carried off. And just so you know, this blood vessel here, it goes to the liver. The liver detoxifies all our blood. So whenever we eat anything, it goes to the liver, and the liver makes sure that, you know, if anything's dangerous, it's removed. So the glucose gets taken away, off it goes. And now let's grab the... I wonder if there's a way to separate it. Copy, more. Nope, sadly. Okay, so let's take this here, my amino acids. So my amino acids, they are small enough to fit. So they go through these pores and they get carried off to the liver. They're gone as well. And then finally, this last one, my nucleotides, they're also small enough to fit through here and they get carried off as well. But the fat, it's not, it's too big. It won't fit. It will not fit into that hole. That hole is too big. It's too big. It won't fit. So what needs to happen is this fat needs to be packaged up in a very special way. And so it gets packaged up in something called by these special proteins, these lipoproteins, and chylomicron. So because these lipids won't fit into the blood, they have to be packaged up slightly differently as well because of the nature of lipids. The lipids are not water soluble. So these lipids, they'll be absorbed into the tissue, but they won't go anywhere until they're packaged up by these special proteins, and we call them lipoproteins. So now I have to, oops, oh yeah, I'll surround it with this. So it gets surrounded by these really special proteins, these lipoproteins that have a combination of lipid and protein. And so this structure, which we call a chylomicron, it's large, right? And it won't fit into the blood vessel that you've seen I've drawn here. So the whole thing will, instead of going into the blood vessel, into the capillary, it will, let's see if I can, nope, it won't cooperate with me. So if I were able to select this somehow, I don't know how to select it, this whole thing would then move into I'll just do this and see if it cooperates. And so this whole thing will move as a chylomicron. It will move into the lacteal, and then it will be taken away with the lymphatic system. And then these fat molecules, the lacteal circulatory system, they are separate. So the red here represents the circulatory system, and it's going to the liver. And this lacteal, it joins the circulatory system in these special ducts that are located around your body. And there's a major one that's kind of around your shoulders, up above your heart. And so a lot of the fat from your diet will get dumped in that region. So these chylomicrons, made up of these lipoproteins, will allow the lipids to be taken away. And that it's necessary because the lipid it's a fat. It doesn't mix with water well, so you need some special mechanism to carry it off. So that is a major part of the digestive process, and that's why I want to do a video for this section. The rest of the digestive process you should be able to pick up very easily from the PowerPoint. But this one here, you might have to play it back and forth, and I'll give you a couple of videos that you can use to help you with the understanding as well, in case this ends up being just too confusing. If you look at this picture the way I have it now, of course, it doesn't make a lot of sense. You do have to follow my logic through, okay? At the end of this process, all the nutrients you need are absorbed in the jejunum, a small intestine. Then the food will continue. Right? So I come back to this thing here. In the small intestine where the majority of the absorption occurred, we get into the large intestine. And in the large intestine, this is where some of our vitamins, our minerals, some of them, and our water is taken up in the large intestine. So that's reabsorbed here. Because we're a terrestrial organism, that water absorption is super important. At the end of the large intestine, we have a rectum, which holds the feces. It's what it's now called, right? The feces. It holds it until a socially acceptable time. Let's see if I can grab this. Yeah. So it holds it till a socially acceptable time, and then any waste that you were not able to digest. The feces is made up of a lot of bacteria, a lot of roughage, and some other things, dead blood cells. These are then eliminated from your body through the anal sphincter. So work your way through the PowerPoint, work your way through the process. I've specifically gone over the chemical digestion that happens in the mouth, the chemical digestion that happens in the stomach, and then the major digestion process that happens in the small intestine thanks to the pancreas, the liver making bile, it's held in the gallbladder. All these things are broken down in the small intestine. And then, of course, the fat digestion is much more complex because it has those additional steps. It moves through the small intestine, eventually the large intestine, the rectum, and done. Okay, so hopefully that's you understand the digestive process. You can work your way through the activity, the worksheet, answering the questions. Notice what's green. We've tried to keep it simple. What is yellow to give you a little bit more to do, and the purple to give you the exemplary piece. As I'll say, like I said last week, if you guys are feeling overwhelmed for whatever reason and you're not able to make your way through the material, that's okay. Let me know. Don't just disappear, because I wonder what happened to you. But let me know. And if you are ready to learn, great. In the PowerPoint, you'll notice that there is a couple of slides. I've been doing this with the grade 12s. I haven't done it with you guys yet. And these slides, I call them optional slides. They're called optional Google Meet slides. So let me just pull one up so you can see what it looks like. In these slides, this is the additional information, the stuff that I find very interesting. And if this were a regular classroom situation, I'd probably take 20 minutes to talk about one of these. But in this distance learning, I got to pull these out. But if you're really interested in knowing about parasites, or really interested in knowing about other things that I didn't include in the lesson proper, I called it an optional Google Meet, which means that you can send me an email, find a friend or two, because it's more interesting generally to talk to 2 or 3 or 4 of you, and then we can meet whenever is convenient for you and me, and then we can talk about these particular concepts. So they're spread all around the PowerPoint. You'll see it says optional Google Meet. It'll typically be purple because they're additional things to elaborate on the material, and even if you see things that are yellow and you want a clarification, that's fine as well. So I'm not making a specific Google Meet. I am giving you the option of meeting with me. Also, I want the menu, if you're doing that, ready for Thursday, and then on Friday, depending on how many people participate, I'll make a time on Friday when we could all meet, and we could talk about some of the meals that you have designed with your meal plan. All right? I think that's it.